does that or help it or what?
They help, right? They help with RNA metabolism, they do RNA splicing and methylation.
Okay, great, oh, they're okay. Then,
They have microRNA, which function can repress, they're much more repress translation, they degrade mRNA, or repress transcription.
so which two functions they do? Tudor family. Do they work in transcription?
Transcription, yeah.
Hmm? Translation. Translation and microRNA degradation, those two functions they perform.
Nothing to do with transcription.
Not the Tudor, microRNA, Tudor family doesn't play with that.
Oh, micro, okay.
Yeah.
I just have that different thing. So microRNA is separate from the two organic.
Yeah. Oh, you guys didn't do any microRNA genetics or anything like that? You only guys did humans.
I don't think so.
They're all the same class.
Yeah.
Yeah. Okay, we did that, I think. Okay, then what happens? So microRNA is a, let me figure it out, just, were you able to read something about that or not? After the lecture last time?
Oh, no,
No? So microRNA are small RNA molecule. They are produced from the genes. So genes are present there. So gene can produce maybe 1,000 base pair long RNA molecule. Those genes have kind of like sequences for microRNA. In a single gene, you can have maybe a gene sequence maybe for 500, 200, or 10 or 1, depending upon. So you can have multiple genes also there for them. So they're produced as a palindromic sequences which are running opposite direction, but they're complementary to each other, okay? And they form the hairpin-like structure. And those hairpins are cleaved off in the nucleus, and they came out from the nucleus in the cytoplasm as a hairpin. And that hairpin is further cleaved by enzyme called lyser into almost 21 to 23 nucleotide base pair. And then they go to then they can do multiple things. So they can go back to nucleus and they can inhibit the transcription depending upon the signal. Or they can stay in the cytoplasm. They can look for a perfectly matched, this is depending upon what kind of signal is there. Perfectly matched mRNA, join that, and then they recruit Argonaut family of protein, and that recruitment is helped by the Tudor family. And then they basically chop off the mRNA. In this way, they destroy the mRNA. Sometimes, actually most of the time what they do, they join to mRNA not with perfect complementary sequence, with some complementary there. Again, the Tudor family help there, these proteins there, and at that time they inhibit the translation. So that actually is the most common way to regulate the concentration of different protein molecules in the cells. And all the development processes, all the disease, actually cancer have so many microRNA dysregulation. So that's kind of like, if a cell needs protein immediately, okay? So what happened, that microRNA will go over there. Another sponge, we did talk about the sponge also, right? So there's another sponge there, which is going to take care of this microRNA, and microRNA will go away from mRNA and translation will begin. Then cells say, hey, I don't need that protein anymore. MicroRNA will come and join the mRNA, stop the kind of switch on, switch off thing. So mRNA degradation means you are basically completely removing that mRNA from. But here, if you want that regulation, on, off, then you need translation repression. So this kind of function is done by another class of microRNA. We don't call them microRNA because they interact with another protein. MicroRNA have Argonate family protein, whereas there is called piRNA, which are in the gonads. They have a PV-clad proteins or MeV-clad protein. They have two proteins. Again, they do the same function. but they are gonad specific. If you have not read this, I will suggest you definitely go and read it on your own. Any question, come and ask me, because this is what whole science is about nowadays. Whole molecular biology is about that. Whole genetics is about that. A lot of new medicines are based on that. And also, if you are planning to appear in GRE or whatever exam you want to appear, there should be several questions based on this. So make sure you are reading it. Okay, then.
Then we started talking more specifically about kiwi. Kiwi is small DNA regulated by transcriptome, and they're a ping pong organ, and they come from the ping pong conjugation group. And then we talked about how Tudor and kiwi inhibit gene transcription. We then talked about BAF proteins, which activate germline nicotine, and then nanos repress translation through different mechanisms and it prevents cell death. The PCG migration occurs in clusters. So then you have the PCGs that are surrounded by other cells that secrete stem cell factors required for survival and motility. They are called traveling niches in the microwave along the sequence path. PCG reaches the genital ridges where gonad development takes place.
Focus, yeah.
So then they can only change to a gonad if DZL protein is present. And the DZL protein regulates mRNA synthesis. With no DZL, the PGCs remain PGCs. And there's a signal from gonad that specifies the timing of meiosis. So then we went to talk about like the male and the female phase.
So what are two signaling molecules there?
WNT4 and
Wnt4. Wnt4. WNT is called Wnt. Wnt, yeah.
Okay, Wnt4 and retinoic acid.
Uh-huh.
And then, so
Okay, then what happens?
So for those meiosis in females, they begin in embryo, and in the male, they begin at puberty. The meiosis of germ cells is regulated by the transcription factor STRA8, and STRA8 promotes DNA synthesis and initiates meiosis. In females, Wnt is made in the developing ovaries, and it also upregulates STRA8 with retinoic acid, which is produced in the developing kidneys. And upregulation of STRA8 induces meiosis, which is called oogenesis in women. In males, STRA8 is downregulated because in the developing testes, the fibroblast growth factor 9 downregulates it, and then the developing kidneys, which make retinoic acid, are stopped by the testes that produce CYP26B1, which degrades retinoic acid, and then no STRA8 means no meiosis. And then we talked about how during gestation, specifically in mice, which is 18 days, at the 13.5 day, CYP26B1 levels decrease, and the developing testes start making nanos2, which blocks STRA8.
Yeah, okay. So now we are going to talk about both spermatogenesis, how the sperms are made, and oogenesis, how the ovum is being made. Okay, so let's talk about spermatogenesis. Spermatogenesis. So spermatogenesis is actually developmental pathway for sperms from primordial germ cells. Okay. So the process of formation of sperm, we said it's a meiotic process, right? Meiosis takes place. And we said in the male gonad, meiosis does not start in the embryo, right? Because of CYP26B1 and FGF9, right? And later nanos. But what happens at the embryo? So the process of spermatogenesis that begins at puberty. Okay, that doesn't occur in embryo. But what happened in embryo? But in embryo. So in embryo, what is happening? That the primordial germ cells we said in the primordial germ cell, they are in the traveling niches and they are kind of reaching the genital ridge. Okay? So the genital ridge where they are reaching, at that time what is happening there, the formation of testes has started. So development is taking place. They are reaching, genital ridges are the places where gonads are going to be formed. Okay? So at that place when they are reaching, they reach there, the testes start forming. And those testes are kind of like, at that time, you see these kind of like tube-like structures are there. So each of these is an individual tube, okay? And these tubes, these tube-like structure are called sex cords. Okay? So, tube-like genital ridges, you have the tube-like structures, and they are called sex cords. Okay? Now, they are starting forming. Now, these primordial germ cells, when they reach there, they are no more called primordial germ cells because they are now going to enter the next phase. Okay? And the primordial germ cell, they enter sex cords. And at that time, these primordial germ cells, when they're entering, they are called gonocytes. So, gonocytes, they enter, gonocytes are actually primordial germ cells, right? But they have now another name. So these gonocytes enter the sex cords. Now these sex cords, when the development is going to take place, when development of testes is going to take place, they are going to change, or they are going to form, not change actually. Where is it? Oh, there it is.
Are you saying the gonocytes are gonna change or the sex?
No, sex cords. Gonocytes are germ cells. They are going to give you sperms, right? Gonocytes are going to give you sperm. The sex cords, when the development of testes is going to form, testes, they form. So sex cords, they form. What we call seminiferous tubules. They form seminiferous tubules. If you look at the testes and the, so this is the cross section of testes. Testes is actually these tubes inside, right? These tubes are called seminiferous tubules. And this is the cross section of that. So essentially, this is a tube-like structure like this, and you have a lumen. So this is the lumen. Okay. Now what has happened? So these gonocytes, they enter sex cords. Okay. Now these sex cords are now developing as the embryo developing, right? They will not remain sex cord. So these sex cords change to what we call seminiferous seminiferous tubule. Okay? So when they are changing to seminiferous tubule, at that time, the level of BMP8 that start increasing in that area. Okay? So what happened? So gonocytes have entered the sex cord. Sex cords are now changing to seminiferous tubule, right? It's a continuous process. And so these gonocytes which have entered there, they are also kind of changing when the environment is changing. We are saying BMP8 levels are going up. And then what happened? These gonocytes, they differentiate into another type of stem cells. Still they are stem cells, but they change into another type of stem cells. And those stem cells are called spermatogonia. So they become spermatogonia. Okay. Now these spermatogonia, the level of BMP8 is increasing more, right? So these spermatogonia, they change to another type of stem cell. So this spermatogonia, level of this thing is there. That changed to another type of stem cell. It is called type AS. This is important. Type AS. S is there. Spermatogonia. Type AS, spermatogonia. Okay. Now these type AS spermatogonia, they are also true stem cells. So what is the characteristics of a true stem cell? True stem cell have the capability to form the full, complete organ system or a particular type of developmental pathway. So here, if you are here in this testis, right? You destroy all the sperms in this testis and you add one type AS spermatogonia cell. And that one type AS spermatogonia cell can repopulate the whole sperm cell population of these testes. Making sense? So that's why they are called true stem cell, because one type AS spermatogonia can enter if you destroy all the sperms here in the developed testes, and one type A spermatogonia there, and that one type A spermatogonia has the capability to repopulate all the sperm population in the testes. That is making sense? Okay, now, what happened? So, right here, this is the epithelia. So this is cross section. This is cross section of embryonic seminiferous tubule. Okay. In that case, what happened? Right there is so this is the epithelium. This is epithelial layer, the outer covering of that. And right there, right here, you have this type A as spermatogonia. Okay? Because these gonocytes have differentiated to spermatogonia, and then that spermatogonia has differentiated into type AS spermatogonia. Right? And then what happened? Right there are the other cells which are surrounding there. right on this side, here, you have another cell. This cell, which is there, is called Sertoli cell. This is Sertoli cell. There are many cells actually. I'm just having one, I just want to show you one type here, spermatogonia, right? There will be so many. And then there is this Sertoli cells are there. Sertoli cells, what they do, they secrete, number one, glial cell derived neurotrophic factor, or in brief you can call it GBNF. Okay? And the other thing they secrete is stem cell factor. We already know about that factor, so I'm writing it SCF. Okay? Now, remember, as the development is taking place, these monocytes are differentiating into spermatogonia. By the time these cells are made, Sertoli cells. Sertoli cells, they start secreting GDNF and SCF. So this transition from spermatogonia to type A S spermatogonia, that is also regulated by the levels of GDNF. So we are saying BMP8 was there, but GDNF also play a very critical role here. Now, the name is glial cell derived neurotrophic factor because this factor was first discovered in the central nervous system, and then they know that this is released by the glial cell and it play a very important role in the development of certain neurons. In Parkinson, the neurons which die, they are all responsive to GDNF. Okay? So, but later they found these cells also make the same neurotrophic factor which is made by the glial. Okay? So now, so far it is okay here? Then, on the other side also, there are another type of cells there. Like here. These cells are called leading cell. Leading cells. Okay. And these cells actually secrete testosterone. They secrete testosterone. And then, right there, you have the blood vessel. So this is the blood vessel. Okay. Now, this structure where you have a type AS spermatogonia surrounded by Sertoli cells, Leydig cells, blood vessel, right? And all these factors are being produced like SCF, GDNF, testosterone. This is called, this whole structural unit like this is called type AS spermatogonia niche. So this whole structure is called type AS spermatogonia niche. Okay, so in human adult male, type AS spermatogonia cells they can produce 1,000 sperms per second. in adult male, 1,000 sperms per second in human adult male. Okay. So what we are saying? We are saying that primordial germ cell, they reach the genital regions. When they reach the genital regions, at that time genital regions, the testis development start, and you see the tube-like structure there, what we call sex codes. These sex codes later are going to make seminiferous tubule in the testes. So they are making those seminiferous tubule in the developing testes. Now, these gonocytes, when they reach there, they enter, while they're entering, they are differentiating into spermatogonia under the influence of high level of BMP8. And when they are reaching inside, by that time, next to, excuse me, next to type A spermatogonia, you have other cells also being developing. So two type of cell I did talk about. I said there are Sertoli cells. Those Sertoli cells make glial cell derived neurotrophic factor and the stem cell factor. Under the influence of BMP8 and GDNF, these spermatogonia, they change to type AS spermatogonia, which are also true stem cells. And we said they can repopulate the whole testis with the new sperms there. And then another cell, the Leydig cells are there, they are making the testosterone. And also some blood vessels are there. So this whole structural assembly is called type A spermatogonia niches. Okay, and we said the type A spermatogonia, they can make 1,000 sperm per second in the egg. Everything is clear here? So just talk to each other for a minute and we'll go further.
That's why, yeah, I just draw it.
Spermatogenesis is development pathways to sperm from PGCs.
So in the embryo, that's what we're talking about now.
Yeah.
So the PGCs reach genital ridge, and the testes are forming tube-like structures called sex cords, right? So when the PGCs enter, they're actually called gonocytes, and those eventually get sperm. So then, then the sex cords start to develop, and the testes form seminiferous tubules, right? Is it after the gonocytes enter, or is that at the same time, or just when after?
I think when the PVCs enter the genital ridge, that starts the testes to make the sex cords, and then I think it develops into that. I think it like changes as all these.
Enters, right?
Yeah.
Like it just goes through the
Like
sex
gonocytes enter
cords.
No, no, no. So PGC, when they
Gonocytes
reach
enter.
there, we call them
Then
gonocytes.
that starts.
Yeah, those develop, and then on like the other side, spermatogonia,
Now monocyte
and then
enter the sex
type
cords,
A.
and then they change to spermatogonia,
And then all these should
So
also
then
have.
we have after this, like, tubules start to develop, the levels of B and P8 start increasing.
We don't call them
in area.
PGC, we call them gonocytes.
Then
PGC
we
reach
have
there, and when they reach there, you start calling them
Simultaneously
gonocytes.
to that, the gonocytes differentiate into
When they
another
get
type of stem
to
cell, the
the site?
Yeah. We
And
name
then
them
into another
gonocytes.
type
Gonocyte
called the
enter
AS.
sex cord. And when the
What is the
monocyte enters sex
GDNF?
cord, so sex cord is then no more sex cord is developing.
We had
Seven
GDNF
phase.
levels, and I just could not
Right?
tell it.
And then-
Yeah, I put it in between this one, sperm and
Making sense?
Virginia and AS.
Yeah, okay.
Okay,
That's a
any
true stem
question?
cell.
Okay, so you're talking, Richard.
So that's a true stem cell, which means like if there's one of them only, it can repopulate it fully developed. And then I go up to my little drawing thing. I have the epithelial thing, the lignin, then this Sertoli cell, the type A cell, the leading cell, and the blood vessel. And so the Sertoli cell secretes the Leydig cell, the GD, the F, and this S
Did you say Leydig or leading?
Leydig, Leydig,
L-E-Y?
L-A-Y,
Okay.
Leydig.
That makes more sense. And then that's called the type AS.
Oh, nice.
And then the last thing I have is an adult male type.
Good question,
So
Rich.
Genesis cell can produce one disease for
What is the frequency?
Right. BMP8 are the other cell in the
So
development.
is this like this cut?
I just provided the niche, the surrounding cells, but they
Cut
are
of one of the two.
There's some, it's a cross-sectional of the embryonic seminid first two, so this one. So like, sex cords, they're tubes, then they form.
Cross-sectional of the. So, do the sex cords form before the PGCs enter?
No, they are already sex cord kind of formation starting when PGC. So
So
PGC,
then-
we don't call them PGC. When they reach there, we call them gonocyte. Gonocyte enter sex cords. Okay.
And then does that start?
Then the sex cord starting forming the seminiferous tubule, right?
Is that because of the?
No,
them
no.
entering or just because?
It's a, say they have to make testes. Their function is to make testes. Testes is made of seminiferous tubule that come from sex cord, right? The purpose of pyramidal germ cell when they reach there, call them gonocyte. Their purpose is to make sperm. So gonocytes are not doing anything to make testes. They are development separate. This is development separate, right? But they are only related because the development of sperm take place in the testes. So the niche of the stem cell is the testes, right? Any other doubt or question here? You're okay? Okay, good. Now, so that happens in the development. Now things are developing. There is no meiosis in the embryo till puberty because the retinoic acid is being degraded, right? So then at puberty, the process of sperm formation takes place. So spermatogenesis has actually 3 phases. Number 1 is called proliferative phase. Proliferation means cell division phase. Okay. Then there is, and when we say proliferation is always mitotic cell division. There is no meiosis in proliferation. Okay. The cell growth is because of the tissue growth or organ growth is because of mitosis. Meiosis doesn't play a role here. Okay. Then you have a meiotic phase. When actually the meiosis takes place, and then you have the third phase, what we call spermiogenesis, when the sperm-like structure is being formed. Okay. Now, how this process starts, then we'll talk about that. We'll cover all the 3 processes. So we have right here the type AS spermatogonia. And this they are right here, right? And then what happened? These type AS spermatogonia, so this is type AS spermatogonia, I'm just running like this, the cell is there. And these type AS spermatogonia, they divide divide asymmetrically. So there are two types of cell division, symmetric and asymmetric. Symmetric cell division is when a cell divides, it gives two types of cells which are similar. Asymmetric is when the cell divides, it gives two different types of stem cells, two different types of cells. Most of the time when the stem cell divide, when the stem cell have to increase their own population, they divide symmetrically. So the stem cell divide, make two stem cells. But when stem cell has to make something else, then stem cell divide asymmetrically. So what is going to have, what is it's going to do is going to do two things. It is going to produce two types of cell. Number one type of cell it is going to produce is type AS spermatogonia. That means it is making a cell that is exactly like this, okay? And the other type of cell it is going to make right here is going to call type A1. Type A1 spermatogonia. Now this cell is going to give you the sperm. Okay? Now what is going to happen? This type A1 spermatogonia, it will divide mitotically. So it will undergo mitosis, and it will divide mitotically 5 times. 5 times. For example, this cell divide like this. One cell give 2 cells. But here is one thing which you can notice here. The cell is not completely divided. It divides but does not separate. There are this cytoplasmic bridge is there. Cytoplasmic bridge. So this is one division. Similarly, 2, 3, 4, 5, right? So it will divide 5 times, and ultimately what you will get so on, right? So you will get cells, so type A1 divide 5 times, and you get cells which are attached to each other, right? So it means essentially, if you see, the nucleus is dividing, but cytoplasm is not dividing, right? That means you can say this is a single cell, actually, right? And each of this cell has many nuclei. So what is happening there? That means there is a formation of syncytium. What is syncytium? Formation. What is syncytium? So syncytium, syncytium is a cell with many nuclei. That's called syncytium. So syncytium formation takes place. Cell with multiple nuclei is called syncytium cell. Okay? Now, so far the mitosis has taken place, right? And these cells which are formed right here after 5 divisions, those are called type B spermatogonia. Now they have become type B spermatogonia. Okay. Now these cells, they divide once. They divide once mitotically. Again, mitosis. Okay. When they divide mitotically, they give another. I'm just drawing like 2, 3, but you can see the number will increase, right? When they divide like this, at that time, after this division, these are called primary spermatocytes. These are called primary spermatocytes. Now what happens? So, remember when I started, I said, okay, the gametogenesis takes place in the gamete, and 2 factors coming from the gamete control those. And I said retinoic acid, right? But later I said retinoic acid is coming from developing kidney. But here, the retinoic acid which was there was taken in the developing testes, was taken care by CYP26B, right? So no retinoic acid there. So then what happens? Now the meiosis has to take place, and meiosis can only take place if you have high STRA8 levels. They should be high, right? And they're high, make them high in retinoic acid. Okay, so what happened that Sertoli cells, what they do, they start making retinoic acid. They start making retinoic acid. Okay. Now, Sertoli cells start making retinoic acid. Retinoic acid, what it does, it makes high level of STRA8, right? So that is happening where? That is happening right at this place. When you have these primary spermatocytes are being formed. Okay? Now, before that, one more thing happened. In this case, where type B spermatocyte, they need to divide mitotically to become primary spermatocyte. So each cell is different. When they're dividing, they're changing their chemistry. They're changing everything inside, right? Because this type B spermatocyte, type B spermatogonia, they cannot undergo meiosis. Only primary spermatocyte can undergo meiosis. And they can undergo meiosis because something had changed during this division. Right? Making sense? So what caused these type B spermatogonia to become primary spermatocyte? 2 things play very important role. They are again secreted by Sertoli cells. Sertoli cell was secreting GDNF and they were secreting SCF. During this process, when the cell are in type B spermatogonia, and at that time the GDNF levels go down. GDNF level go down. And SCF level are high. Okay. When that happens, type B spermatogonia divide to give you primary spermatocytes. That is, everyone is getting it? So what is happening here actually? So these are your Sertoli cells like this. So this is Sertoli cell, right? This was the first one that was type AS, right? Then they are all dividing. Ultimately what you are getting here is, this is your type B. And then you have your primary spermatocyte here. And right here will be the lumen. This is the lumen. So you can see here you have to have high GDNF and SCF. Remember to maintain those. So you can see this is the anatomy. As you're going away from here, it means right here the Sertoli cell is secreting less GDNF than it was secreting very high GDNF here. Making sense? Same way, it is secreting something here which is not secreting there. And what was that? We said retinoic acid. Right? So retinoic acid is secreted right here to the primary spermatocyte. So they're like this, going like this. And then right here we are saying that retinoic acid is secreted, so that means the high level of STRA8 are here. So that means right here you are going to have the first meiotic division. First meiotic division is going to take place. And when the first meiotic division are going to take place, again you are going to have these cells. These cells will be again like this. First meiotic division has taken place. And now these cells are going to be called secondary spermatocytes. Secondary spermatocytes. And then after first meiotic division, you will have because meiosis have two divisions, first meiotic division, second meiotic division, right? Then the second meiotic division is going to take place. Second meiotic division, and then you will get again. So for example, there were 4 cells, now it becomes 6 like this. I'm just drawing it. And these, which are formed here, these cells will be called spermatids. Spermatid. So these are spermatids formed. So, right here you will have spermatids. Okay? So this is how these spermatids are very close to lumen now. That is making sense? So we started with type AS, A1, and going from here we are coming here to the spermatid which are right near the lumen. Now, so what control the concentration of GDNF and SCF? We are saying the concentration goes up, goes down. What control? Right? So there is a hormone called follicular stimulating hormone. That hormone is coming from the developing pituitary. Follicular stimulating hormone control GDNF and SCF concentration. So, what happened? We are saying we get the spermatids. Their nuclei will be haploid or diploid. Haploid, because undergo meiosis, right? But technically they are not haploid, because whatever this nucleus is making is being shared by this cell, so they are still attached to each other, right? So they are apparently haploid, but technically they are still not haploid. So what happened there? Actually, if you think from here, from this process to this process, the differentiation has taken place. What has happened? Differentiation. What means differentiation? Means the differentiation cell has differentiated to something. Here it was a pure stem cell, that stem cell started dividing, give you type AS changes type A1 divided, changes its morphology, changes its chemistry, that gave type B then changes, give you primary spermatocyte, secondary spermatocyte, and you get spermate, right? So the differentiation has taken place throughout. So proliferative phase. So type A stem cells, they divided, increases their number. Then they divided asymmetrically, give type A1. Then type A1 divided, increases number. Then type A1 divided, give B, increases number. And then B divided, give primary spermatocyte. Proliferation has taken place. All mitosis was there, right? And after the mitosis, so the proliferation phase took place, then you get the mitosis, which happened because the steroid level goes up because the Sertoli cells started making retinoic acid, right? And then there the meiosis took place, and then the result of meiosis was formation of haploid nuclei. You get the spermatids there. Making sense? So let's go back. We are saying the spermatogenesis have 4 phases: proliferative phases, meiotic phase, and then spermatogenesis. We didn't talk about spermatogenesis yet, okay? So what happened, we are saying that we have this type AS spermatogonia cell, which was attached right here to the epithelia, that divide mitotically but asymmetric division, gave one type A spermatogonia, make more of those cells, again stem cell, and give type A1 spermatogonia. That type A1 spermatogonia divide mitotically 5 times, and when it divide, it give you a type B spermatogonia. They are all attached by cytoplasmic here.
So I have a question. So it divides into the new one,
Yeah.
the type A1. So then it does the mitosis 5 times. Do all of those stay connected as they get closer to the
Yes,
lumen?
yes, they're all
Okay,
connected.
So it's like the type A1 is connected to the next one, and then that's connected to.
Oh, yeah,
the
they
first.
are connected, say it again. They are connecting to each other, not connecting to previous one.
Okay.
This is not connected to this.
Okay.
This is connected to this.
But it's not connected to the one.
No, they are not connected to each other.
Okay.
They're not, they're just, this cell is here next to this cell is here next to this is this cell.
Okay.
But there's no connection. Okay? Now, primary spermatocyte is formed. We said this transition of type B formation is controlled by GDNF and SCF. GDNF level goes down, and then that tells these cells to divide mitotically and become primary spermatocyte. Okay, and SCF level stay the same. And then we are saying that these primary spermatocyte, because of the level of retinoic acid going up, steroid going up, they divide once mitotically, divide mitotically, first meiotic division take place, we call them secondary spermatocyte. And then the second meiotic division take place, and they call spermatids. So in this way, from one cell, you get 4 cells undergoing meiosis, right? And you get 4 haploid nuclei there, but they are still attached by cytoplasmic bridges, so technically they are still not haploid. Morphologically, they look haploid. So far, everything is okay. So talk to each other about this process and we'll go further.
The phases are proliferation phase, meiotic phase, and then spermatogenesis. So I haven't talked about spermatogenesis. So then type A is divide asymmetrically.
Before that, I forgot to mention one more. There is a mutation called Luxoid mutation. Luxoid, Luxoid, Luxoid mutation. In this mutation, what happened? This mutation takes place, and all the males are sterile. So male, all sterile. So in this case, what happened? That the regulation of type B spermatogonia, it is regulated by a transcription factor. All are transcription factor, right? But that mutation caused the problem in the transcription factor that regulate the type B spermatogonia division. And because of that, what happened, all of these type B spermatogonia which are formed at once in one time, they all change to meiosis, everything becomes sperm, and all the sperm are made in one time. What that does, that actually clears off all the stem cells. No more stem cell is left in the testes. So all the sperms form one time, no more stem cell left, and these animals become sterile. Making sense? Now, we said all these cells, they are responsible to the GDNF, right? So GDNF is a trophic factor. It means that these cells should have a receptor, right? Here should be a receptor to which the GDNF can come and attach, right? So only then they can work. Remember when first time I was talking about my research, I said we work on the Hirschsprung disease, the megacolon, right, in which the neuron cannot migrate down the gut. So what happened? Many of the cases of the Hirschsprung disease, they have a mutation in the RET receptor. So people who have that disease, actually we'll talk about kidney also. Kidney also, RET gene play very well. So those people who have Hirschsprung, they have abnormal testicular development. Kidneys, they don't have no kidney. We made some knockout mouse, we knocked down RET. Those mice die even in 24 hours because no kidney. Of course, male will not have testes properly made, but so you can see this is a very, very critical receptor, and trophic factor plays so many things. What we talk
So
more
it's a
about.
GDNF receptor isn't.
GDNF is the trophic factor, right? Receptor is RET, R-E-T.
Oh,
Receptor is called RET, R-E-T. GDNF is the factor which binds to receptor for RET. Okay, now you can go and talk about it.
Was that different than the looks one?
so derived from type AS, which is the same as type A1. So type A1 is going to be the one that produces sperm. Like there's pretty much doses 5 times, right?
Oh, so you have to forget that they have been right.
And this is the formation of this one that is that's what it's called. The multiple.
Mm-hmm. So, yeah.
I do have.
And then it divides one meiotically,
Yes, yeah.
and this is called the primary one, primary phase. And then this can take place with high levels of strati, so serotonin cells start making retinoic acid, so those levels can increase. So before that, the serotonin cells are secreting GD, NF, and SCF, so the levels of GDNF go down and the levels of SCF go up. And when this happens, type B can become primary.
Yeah.
So then the primaries are made, and then that causes the first mitotic division to form secondary mitotic
So when the Sertoli cells start
and then the secondary
secreting retinoic acid, which causes striae to go up.
which are apparently
That then
haploid,
allows
but they're
the
still
first
connected, so they're not
meiotic
haploid.
division.
Yes, meiosis.
We have something about the hormone.
So meiosis is two
The
steps.
follicular
Meiosis,
stimulating
second
hormone,
meiosis.
which controls the
The
levels
start
of
very
GD
good.
and F and
Start means
SCF.
you have to have
And it's from the
meiosis.
developer from the pituitary. And then we have that drawing
There's
that
the secondary
which is
steps.
Yeah.
So timeline,
I don't know what the left is. It doesn't mean the left is okay to the form.
Was that how it looks like?
Yeah.
I thought you said it was like H1 or something. So,
Can you talk to us about the Luxor mutation?
So Luxide mutation is mutation in the transcription factor which regulate the division of type B spermatogonia, right? So when that doesn't happen, all of the stem cells, they basically, all the type B change to sperm. When all the type B change to sperm, that means stem cell think, hey, we need to make more. So they are exhausted, so no more stem cell is left there. So all the sperm are formed at one time. So the testes are completely devoid of any stem cells, so they cannot make any sperm anymore.
So what was the thing about the red receptor?
Oh yeah, so I was saying that it means that when you have the GDNF, there should be receptor, right? So GDNF is excluded, so all these cells have the receptor for the GDNF called red. So it bind and then bring the changes. So I just wanted to draw the attention that this system is critical for many developmental processes, including, of course, testes, but nervous system, kidney. So, and I gave the example of the Parkinson. I said in Parkinson, the neuron in substantia nigra, they die. They are all dependent on the red. And people thought long time ago that maybe the therapy with GDNF, giving them the GDNF might help. And they made some viruses actually which they injected in the brain, and then that was making GDNF for them. And they found like it was not that successful because the way the neuron die is very different from other cell type. So, but there was partial success. So that's why I brought that in the discussion.
yeah.
Doesn't the pituitary secrete follicles stimulate?
Say again?
What secretes the follicle stimulate?
Which hormone?
This one.
Follicle, pituitary.
Pituitary.
Pituitary, yeah. Follicle, pituitary. Any question other than that? You guys are okay here?
Yeah.
Okay, you guys are fine? Okay, then we are talking about spermiogenesis. Spermiogenesis.
We have 3 minutes left, so do you want?
Oh, we have 3 minutes left? Oh, we will not be able to finish it. Okay. So that's okay. So we'll continue with this on Monday.